And today we're speaking with Dr. Hashem Koohy coming to us from Oxford. His talk is going to be towards decoding the principles of antigen-specific T-cell response in time and space. Just a little bit about Dr. Koohy. His current position is Professor of Systems Immunology, MRC Human Immunology Unit at the Oxford University. He's also the program leader in MRC Human Immunology Unit. His research theme is taking multidisciplinary approaches at the interface of immunology and data science in order to decipher the rules underpinning T cells' interaction with target antigens. He was educated at Warwick University. He was a postdoc at the Sanger Institute in Cambridge. He also was a postdoc at the Babraham Institute in Cambridge. And other professional roles and contributions are the Alan Turing Fellow, member of the advisory board for the F1000 Research and Applications of AI and Health Gateway, associate editor of Frontiers in Immunology, and a member of the editorial board of Immunoinformatics. Welcome, Dr. Koohy. Thank you very much for joining us.
Thank you so much for the invitation.
Yeah. So what are we diving into here today?
Yes, yeah, I can do that. Yeah, thank you again for the invitation, and it's really a pleasure to be here and to share with you some of ongoing works in our group. And because majority of what I'll be telling you are under revision, I'll be very keen to hear your opinions and thoughts about that. And I would like to start by apologizing because perhaps the title you see right now is slightly different from what you saw in the flyer. And the reason for this is that I thought if I put it in the context of what's going on in our group, then the whole thing will make more sense. So but I'll promise to cover enough of what you saw in the flyer. So we all know that basically T-cell interaction with pathogens is really at the core of a number of serious human diseases, and because of that, it has basically attracted a great deal of attention both in experimental scientists and also in computational community. In experimental side, if you have a given antigen or actually a multiplex pool of antigens and HLA molecules, these days it's fairly easy to pick up a single cell technology to basically profile for target TCRs. And in addition, you can get transcriptomic, proteomic, and even epigenetic of that. But the pressing issue here, among other problems, is that this type of technologies really work with those known antigens. And most of the time, the prime suspect is an unknown antigen. And therefore, for those situations, we cannot use this type of technologies. On the other hand, if we have a T cell or T cell receptor of interest, then we can use one of these so-called combinatorial yeast display libraries to basically scan this TCR versus literally billions of peptide MHC complexes. And at the end of the day, we will be left with a pool of a handful of basically targets for that given TCRs that then we can actually take some additional computational approaches to learn the pattern of T-cell cross-reactivity and identify future targets for that T-cell. But the problem here is that we end up doing one TCR at a time, and these technologies are not scalable to single cell technologies as they are now. And in the computational side, if you have some labeled TCRs with known antigens, you can pick up one of these basically supervised predictive models, try to predict what peptide MHC complexes they are likely to recognize. If you have orphan TCRs, on the other hand, you can again go for unsupervised clustering. And the idea here is to basically cluster TCRs based on their so-called common specificity, and then learn the sequence footprint of each of these common specificity groups in order to predict future members of each of these groups here. But the problem again here is that for instance, for supervised clustering models, we and others have shown that at least for those TCRs with unknown antigens, they do not really do much a better job compared to a simple, say, Hamming distance that you will be comparing to sequences based on the number of amino acids that they differ. And for predictive models, if we want to apply them for unseen antigens, as it has been shown in this preprint, they are not actually getting much better than random predictions. And therefore, this whole thing suggests that there is actually a big gap in the field. And this is basically where we work on this topic. And if we look for the roots of the problem, most of the time in textbooks, the diversity at immune repertoire or at the level of peptide MHC complexes are basically highlighted as key factors of this complexity. But I would like to think that actually this diversity is the symptom, is not a disease. The real problem is that T-cell interaction with pathogens are really fuzzy, is very sloppy, and there are quite a lot of things going on. Two key components are T-cell cross-reactivity and common specificity, and these actually root back in timeless positive and negative selection. And therefore, if we want to really understand this interaction, we have to study these two key components in parallel. And in addition, we have to put it in the context and leverage other information that we get these days from single cell technologies in order to make a better sense of it. And for today, I'll be mainly focusing on T-cell cross-reactivity. And if we just pick up as one example, targets of for a cross-reactive T-cell and look into their sequence footprint, we see that in addition to anchor position, in TCR contact position, we see very conserved motifs. If actually we put targets for a bunch of TCRs, target peptides for a bunch of TCRs into a clustering algorithm, we see that they actually cluster fairly well based on the TCRs. And that by itself suggests that if we have enough data of these T-cell sequences and their target peptides, it would be possible to link these two with each other. But the problem is that we don't have that much data of this characteristic. And therefore, we need to generalize our assumption a little bit and go beyond cross-reactivity and look into features of peptide immunogenicity. So here we don't care which TCR.
Hashem, can I interrupt you for 1 second? We just have a question in the chat.
Yeah.
The question is this all for class 1 MHC?
Yeah, mainly I will be focusing on class 1 on CD8 T cell. Yes, that's a very good question. Yeah, sorry.
Okay, great. Thank you.
Yeah. So here by immunogenic, I refer to those peptides that, based on one of those functional assays have been labeled to have basically target for a T cell response. And if we look at TCR contact positions of these peptides, again, we see that there are some peptides which are enriched in TCR contact position of immunogenic peptides, whereas we see some of them are depleted. And signals like that has attracted the community to develop models to predict immunogenic peptides or targets for CD8 T cells. And at the beginning of pandemic, we were keen, actually before pandemic, we were keen to use those models to predict neoepitopes for cancer. But upon emergence of SARS-CoV-2, we were also keen to use them to predict targets from SARS-CoV-2 protein sequences. And we realized that actually they do not do much better than random predictions. And for cancer is slightly better, but still is far from perfect. And then we decided to look into the roots of these problems. The very first one is that we realized that the correlates of immunogenicity is not universal among cancer neoepitopes and viral peptides. The second one is something that we have labeled as HLA bias or inappropriate integration of cross-HLA variation. And what I mean by that, if you look into, as just one example, if you look into the distribution of positive and negative among these HLAs, you see that positive are mainly dominated in HLA-A201, and the negatives one are basically dominated in the rest, for which we have enough number of peptides. And by seeing this, it's not very unusual to assume that perhaps this model will be picking up HLA instead of predicting immunogenicity. And in fact, when we tested this, we realized that actually this is true. And this is not just specific to this example. If we are not careful, we all know that all peptides that are presented by various HLAs, they have very conserved motifs at their HLA contact positions. That can be seen here, or from the full-length peptides can be seen here. And also we previously shown the same in the self-peptides. And therefore, if we are not careful, then models will end up picking up these conserved per HLA signals and not predicting the peptide immunogenicity. And the third issue is something that Koohy, who developed this her model, labeled as out of distribution problem. And what we mean by that is that, so if we put all the peptides in one bag and just randomly shuffle them, pick 90% as a train and the rest 10% as a test, and then look at the sequence footprint at TCR contact position, we see fairly similar pattern here. However, if we put everything non-SARS-CoV-2 as a train and use SARS-CoV-2 as a test, and then look at the sequence footprint, we see a fairly distinct pattern here. And that is not specific to SARS-CoV-2. We also see for other viruses that we have enough data. And for that reason, if we take a usual 10-fold cross-validation, the model picks up fairly well. But if we go per virus, then they actually fail accurate prediction. So Koohy decided to address these issues and some other issues that I don't actually explain here because of the time. And she developed a model, and then she investigated its application for predicting cancer neoantigens and also viral epitopes. So what is novel about her model? Firstly, she basically utilizes transfer learning at the embedding stage. This is one thing that is fairly distinct. And the second one is that the model has two key components. The first component takes an HLA agonistic approach to deal with HLA bias. And because of this model actually works better for in data sets where we have imbalances of immunogenic across different HLA types. And we all know that a T-cell response is very HLA restricted. Therefore, we have this second component here that take actually care of this. And also here she introduces a module to take care of out of distribution. And TRAP is also very flexible in a sense that the user can get some high confidence prediction that are good enough to go ahead and do some experiments if you want to. And for low confidence, then depending on your predicting cancer neoepitopes or pathogenic, you can take different strategies. And there is another novel modular strategy here. This is a relative similarity to autoantigen and tumor-associated antigens. And here you can think of this as an analog to, in cancer, we have wild type and mutant that most of the time they differ one amino acid with each other. But when it comes to viral peptides, it's difficult to basically find the human counterpart of that. And she uses this module to take care of that. And then she basically checks model performance based on different transfer learning approaches or different architectures, and see fairly reasonable performance. And she applies the model on predicting cancer neoepitopes from in-house GBM patients, the data that we have from GBM patients, using the default parameter, and then applying some of the new modules that she has implemented in the model, and increases the prediction accuracy of the prediction. But I would like to actually take a little bit more time to explain the second application, which is basically in a companion manuscript which is under revision. And here we have been investigating the impact of mutation on CD8 T cell target on T cell response. So here the story or the assumption is that we have this wild type from Wuhan. We have collected all those peptides. And there are two scenarios. One, that in Omicron, they are not mutated, they remain as the same. And the second is that those are mutated in subvariants of Omicron. And then for this category, for this mutated, we have three possibilities. Both wild type and mutant are presented by a given HLA. Wild type is presented by HLA, mutant is not, and the other way around. And then this is implemented into a mathematical model that has two key components. One, A, is basically the probability of presentation by HLA, and the second is T-cell recognition potential that we get from TRAP. And then once we do some exploratory analysis to make sure that we are in the right direction, then we take in additional mutagenesis simulation to create a combinatorial space of all possible mutations for each of these peptide at each position for each possible amino acid. And then we implement kind of clustering to kind of reflect the different escape trajectories or different escape routes at each position. So just a few exploratory figures to make sure that we are in the right direction. As we look into the presentation score, we see that there is a slightly impairment in Omicron compared to Wuhan. But if we actually break down into super HLA types, we see that the most deleteriously impacted is those spike drive peptides at a BO7 super type. And among these, we see these peptides that they have this so-called basically this peptide, which is in actually, this controversial super antigen peptide. So I don't have actually anything to do with that. But what I'm saying here is that this is basically the peptide that overlaps this motif here. And the point here is that we see two HLA types, both from HLA BO7 supertype are the most basically impaired ones compared to Wuhan. And if we look into other peptide MHC complexes that contain this peptide, we have altogether 15 of them, and 9 of them are actually uncoupled compared to Wuhan, meaning that actually T-cell response is left for this few HLA types among these complexes. And in fact, we see about 9 of Wuhan peptides upon Omicron, they become completely invisible to the T cells, CD8 T cells. And then we start looking into TRAP. First, make sure that TRAP picks up Wuhan CD8 T cell targets, which is also reflected here, and then look at the final immunogenicity score. We see a slight difference, a slightly lower immunogenicity, which is consistent with what other people are talking, and actually is better reflected here, kind of similar immunogenicity. However, we have a few peptides from Wuhan that have been basically affected upon mutation in Omicron. And then in mutagenesis, because of the time, I'll actually just skip that slide. So what we see here basically is that when we look into the combinatorial space, we see either removing or imputing amino acids based on their physicochemical properties actually is affecting the immunogenicity scores. So if you remove, then you lose immunogenicity. If you impute hydrophobic or polar amino acids, you increase the immunogenicity. These are at just amino acid levels. If you also take the peptide position into account, then you see a more clearer picture. However, perhaps the most interesting pattern is seen here. This in x-axis, you see Wuhan CD8 T cell target from functional assays. And in y-axis, we look into either T cell recognition potential, or equally, you can think of T cell escape potential, because these two mirror each other. And we see a very, dynamic pattern. However, three categories here can be highlighted. Those that upon mutation or in this space, they have enhanced immunogenicity overall. The other ones are these that impaired immunogenicity, and some they have tolerant to the mutation. And in a sense, the way we would like to think about that is that upon mutation, these are more secure for T cell response, and these guys here will be actually potential for T cell escape, T cell immunity escape. And if we look at actually what is contributing to this pattern, what we see is that, again, the fraction of hydrophobicity in the wild type is actually one contributing factor, and also the number of HLAs presenting each of these peptide is another contributing factor. And just for the last two or three slides, I'll show you a couple of specific examples to help making sense of what I was arguing here. So if we look at this specific mutation here in wild type and in the Omicron, here we have 10 clusters, each reflecting basically escape potential at a specific position. And the red or yellowish is actually kind of reflective of that the mutations are more immunogenic, or in a sense that the escape route is closed, because the mutation will be immunogenic. But the bluish will be kind of reflective of this is actually an open escape potential. This particular peptide actually has a mutation at position 7. And another thing to note is that here you see that the majority of routes are closed, immunogenic, and therefore this is among those enhanced that I showed you in previous figure. And also this controversial peptide is basically pretty much all routes are closed, but the route 2 is open. And interestingly, the virus has given actually rise to a mutation at this trajectory or this route. Whether that is a random or it's under T cell pressure, we don't know, because 1 out of 9 chances that this mutation to take place here. And the third is among those that we had impaired immune response, and you see that majority of routes are open, they are blue, non-immunogenic. But what we see here, the virus has given rise at position 7 or at cluster 7. And among this cluster is actually this one here, which is one of the most deleterious mutation that can actually happen at this position. And basically, if I want to wrap up here, is that we believe that there is a knowledge gap in basically understanding T cell interaction with pathogens. And towards this, we developed a trap to basically help us identify CD8 T cell targets. And we have used trap in 2 different contexts, for cancer and for predicting CD8 T cell targets and the impact of mutation on T cell response. And I'll be happy to take any question.
Thank you very much. Yeah, this is a very interesting approach that you guys have developed. I wonder if you might comment on how TRAP might be used in context of autoimmune diseases like type 1 diabetes. I know we spoke earlier and you said that's not your specialty, but when we're looking at that tool, it looks, there's, the whole idea is, the beta cell undergoing suicide or murder, right? In the on the pancreas. So if it's contributing to its own demise, perhaps it is sending up this HLA MHC1, sorry. And so maybe then this might be applicable, correct?
Yes, that is true. However, I should admit that actually, in the context of autoimmune diseases, it's more complex because at the end of the day, you will be basically trying to find some CD8 T cell targets from self protein, basically.
Mm-hmm.
And that is not something that we have directly implemented or considered in this model. And actually, in one or two cases that we have come across, for instance, in autoinflammatory diseases, we took a completely different approach. And what we did, basically, we started with some TCR that we detected them in inflamed tissue, and we could see some basically clonal expansion and some pro-inflammatory signatures. And then we basically use that yeast combinatorial approach that I showed at the beginning to basically profile all the possible targets for the given TCRs. And then once we have a pool of targets for that given TCR, then we can actually take that as an input for a machine learning or computational tool to scan the whole human protein sequences and find out additional cross-reactive targets for that given TCR.
Okay, yeah, that's very interesting. Do you know anyone in that's currently using this kind of approach in context of autoimmunity outside your group?
Yeah, we haven't, as used as such, because it will be a little bit difficult, as I said, and not considering
Right, you just sort of
the
stumbled
underlying
across.
immunology in the model development. So I would say that just directly picking up other model or a similar model. However, basically, it will be good to have a conversation with Chloe. And Chloe developed this our SAT module here. And when she was developing this module, she was basically spending quite a lot of time on understanding response to self-antigens by looking, so this module here, by basically comparing this to those self-peptides that are presented in thymus, I believe, as a control.
Mm-hmm. Yeah, that makes
So
sense.
yeah, I think I need to either ask her or or perhaps we should ask directly from Chloe to see what she thinks about that.
Yeah. Okay, maybe she's going to be the next guest,
Yeah.
future guest.
Thank you.
It is a very interesting system, and it's, obviously has loads of complexity, but it's really cool how you used the SARS-CoV-2 to really kind of test, I guess, the model.
Yeah, it's it's still I would admit that it's far from perfect, and honestly speaking, if we see a model claiming that this model can predict immunogenic peptide with very high accuracy, 90% or above, then I wouldn't buy it easily, because I have seen the complexity behind this process. But in this model, what we tried to do was to basically tailor it with some real immunological basically foundation for that, to make it immunologically make sense.
Yeah. I'll open it up to anyone in the audience. If someone would like to ask a question, please unmute yourself and ask a question. Anyone? Okay, silence. Feel free to reach out. Feel free to reach out to Dr. Koohy if you have further questions. You can connect with him at I believe, let's see, at Oxford. You could read this paper and find him in the rolling credits, or probably at via LinkedIn. So thank you again, Dr. Koohy, for sharing these data with us. It was really fascinating, and wish you all the best. Have a
Not a
great
problem. Yeah. I'll thank you all. Thank you for invitation, and you're absolutely right. I'll be delighted to basically discuss any question you might have, or any actually suggestion, or any comments that you might have. So please feel free to reach out, either myself or any group member, Chloe in particular, who developed this model.
Fantastic. Thank you again, and have a great rest of your evening in England.